All right, guys, I know you just did your DNA exploration activity and it was going over the basics of DNA. And as you learned, you have the bases A, C, G, and T inside DNA. And DNA is found inside the nucleus and can only be inside the nucleus because DNA is such an important molecule. We don't want to take the chances of it getting damaged. Now your DNA, yes, it's made up of sugar and phosphate base backbone, sorry guys, but the bases are the most important part. This is actually the gene, and the gene is what codes for a specific trait. And today we're going to talk about how this gene can be actually used to create a protein. Now before we can actually do that, we need to talk a little bit about the different types of RNA. Now you learned RNA has A, U, C, and G. And if you notice, that's very, very similar to DNA. However, DNA doesn't have this U, so that's what makes it different. In DNA, we said that A pairs with T and C pairs with G. But in RNA, we don't have that T, so we use that U instead. We replace that T. So there are two types of RNA, which you're learning through this protein synthesis computer activity. The first one is tRNA. tRNA is called transfer RNA. And it's called transfer RNA because it does exactly that. It transfers over amino acids. Well, why would it need to transfer over amino acids, you ask? Duh, those are the building blocks to make proteins. And that's exactly what we're making. Now this tRNA is going to carry these amino acids to the ribosomes. Why the ribosomes, you ask? Because that's where proteins are made. So what is the structure of tRNA? Well, this is kind of like how they depict it. They make it look kind of like a T or a cross, and they have 3 bases. And since this is RNA, you'll never find that T. You'll find either A, U, C, or G. But T, you can think 3 bases. T, 3, TT. And on the other side of this, you'll have the amino acid. So right here is the amino acid. This is the tRNA. And these 3 bases are called an anticodon. The other type of RNA is called mRNA. And this is messenger RNA. Now, as you learned from DNA exploration activity, DNA is such an important molecule that it can't leave the nucleus. So mRNA is its best buddy. It comes over and copies DNA. It gets all the information from DNA and takes that information and goes to the ribosomes. Now, if you notice, mRNA has many bases because it's copying DNA, which has many bases. Many, mRNA. So now here I have a molecule. Take a look at this molecule. What is this molecule? Did you think in your head DNA? If you did, you are correct because here you have T's and you have no U's. Well, we need to transcribe this into mRNA. So we need to write down the complements. Now A goes with U in RNA, not T's. C goes with G. G goes with C. C, G, G, C. And if you notice, I'm just continuing the trend of writing down their complementary base pair. If you also notice, there's no T's in here, just U's. Here now I have many bases, so this is messenger RNA. Now, messenger RNA in these many bases have secret codes that are going to code for our amino acid. Similar to our words, we have C-A-T. When you see the word C-A-T, you think, meow, cat. But if I have T-A-C, now you're thinking, ouch, that's a tack. You know me and my drawings. So that is a totally different meaning from cat. And we have some words that are as small as one word letter, or words that are as long as, well, who knows, 15 letters. In RNA language, they're always separated by 3. So here I have 1, 2, 3, split, 1, 2, 3, split, 1, 2, 3, split. And each of these 3 are called a codon. And they're going to code for a very special amino acid. Let's talk about how that gets done. So now here I have my gene again. I'm going to first transcribe this into messenger RNA. And this occurs inside the nucleus since DNA is only found inside the nucleus. A goes with U. C goes with G. C, G, C, G, U, G, G, A, U. So here I have now my messenger RNA. Now mRNA, we said RNA, it's single and likes to mingle, so this single-stranded molecule now is going to go to the ribosome. And the ribosome is going to read these codons, the 3 bases at a time, one set at a time. So here, a ribosome looks kind of like a misshaped snowman. That's the ribosome. It's like an old typewriter. It takes only in a little bit at a time. Now tRNA has that anticodon, and the anticodon is going to match up with the codon. So if I have U is going to pair with A, G goes with C, and C goes with G. So over here I'm going to find my anticodon ACG, and here it is. I have my ACG, I have my tRNA, and over here, what's this fancy thing? You're right, an amino acid. The next thing it's going to do is I have my next codon. I have GCG. So I'm going to look for the anticodon CGC. So I found my anticodon CGC, which these tRNAs just are floating around in the cytoplasm, and then they come and match up, the anticodons to the codons. Once they match up and the tRNA drops off the amino acid, we're going to have a nice little tiny bond formed between these amino acids. Try thinking back. What is that bond between the amino acids called? Correct. The bond between these amino acids is called a peptide bond. And it holds the amino acids together. Now, if you notice, I took away this tRNA. Similar to the male person, when a male person drops off a package, they don't stay and hang out with you forever. They go and pick up a new amino acid. So this tRNA, which doesn't have an amino acid anymore, is going to go pick up a new amino acid from the food that you digested. All right, now I have UGG. So I'm going to look for ACC. ACC comes over. You're going to form that peptide bond. This tRNA now can leave. Now I have AAU, so I have UUA come over. I form that peptide bond. This tRNA leaves. Eventually there'll be a codon that codes for a stop, and now I have this molecule, a sequence of amino acids, and that is a protein, or if you remember, a polypeptide. All right, one last thing I want to cover before I let you guys go is that you're going to see this fancy chart during the Regents exam and possibly on your tests. And we're going to start again looking at this molecule. What is this molecule? Did you say DNA? If you did, you're correct because it has the T's there. We need to first transcribe DNA into mRNA. So we can transcribe DNA into mRNA in the nucleus. A goes with U. C, G, G, C. Continuing down. And now I have my mRNA. How many bases are inside a codon? Correct, 3. You split it up into 3s. Now you're going to use this chart to determine what amino acid should be placed. My first letter is U. So I look over here where it says first base and I find U. My second letter here is G. So I go to my second base and find G. Now I want to see where these two meet, and if you notice it says UGC, so I look for where it says UGC and it says CYS. My first amino acid is CYS. Doing it again, I have G, I find my G. Now I have my C, I found my C, see where they meet, GCG, GCG codes for ALA. I draw my line, which represents my peptide bond in between those two amino acids. All right, UGG. U, G, UGG, TRP. TRP, my trusty peptide bond. And our last one, AAU, A, see where they meet, AAU, ASN, ASN. So now I have my amino acid sequence, or my protein, or polypeptide. I hope this helped you understand what we were talking about during the protein synthesis computer lab. Good job, guys.